I don't forget. All right, listen carefully. Your first lab reports are due today. Blackboard lets you know if they're on time or not. Everybody that's here completed the syllabus assignment successfully, so you should know the right from wrong. That's simple. Okay, second, starting with this lab, you're going to have to start creating graphs. So write this down. When you read a question, if it tells you to create a scatter plot, that is a graph. Okay. What I found is I have students that take this course and they're clueless as to what a scatter plot is, and you're not supposed to be. Okay? You're not supposed to be. In either a previous biology class or definitely in chem, you should have been introduced. But just write this down now. I'm trying to help you out. If you're asked to create a scatter plot, which you will be asked to create a few, they're saying create a graph. On that note, I use Excel. Some of my students, will use other programs such as Google. I think, what is it, the Google Docs and all that. But the thing is, the reason why I use Excel is because you're also going to do a statistical analysis of your graph, where it's going to ask you to do a regression. It's basically asking you to create what my son used to call in high school a regression graph. It'll ask you to create a scatter plot, usually using Excel. Then it'll ask you to do a linear regression analysis, a best fit line. Okay, and then it'll say, hey, look, ask it to give you the linear regression equation, y equals mx plus b, and sometimes it'll ask you to give the r value. Okay, and I'll explain what all that is. So you have to become familiar with this. Now, there's been a recent turn of events. If you're like me, maybe on your computer, starting, I believe it was yesterday, anytime you open up a Word doc or a PowerPoint or Excel, you get a message that this program, this version is not going to be supported much longer. What's happening is everybody's being pushed to use the more recent versions of Word, Excel, and PowerPoint. Okay, and if you don't update your program, you're going to be out of luck. So listen carefully. ACC has done what they can to take care of this. I'm going to post a link under syllabus, and I'm going to title it Office 365. When you click on that link, it's going to take you to an ACC page. Read that page. And what it does is it tells you how you can upload Office 365 to your computer. Okay, so that you don't run into the disaster that's going to happen for many at the end of the month. It is free for students and ACC employees. Okay, so I'll post that link after class under syllabus. Take a look at it, please, okay? All right, because I had a student already ask me, sir, how am I going to create graphs? I don't have Excel, I'm using a Mac. No, I've had students use Macs and PCs. You got to figure it out. It's an online class. Okay, I know it sounds harsh, but I went over all that on the first day, everybody. And trust me, I'm struggling too. I didn't like that message when it popped up yesterday. Okay. Yeah, the only problem is, Caleb, with that is they had some students tell me they can't do the regression analysis. I don't know how true that is anymore. If you can, then great. Yeah, Google Sheets. That's what I was trying to think of. I said Docs, but Sheets. Yeah, you may be able to. There's also an app. I don't know. See what my friend said. Trying to find it real quick here. Am I pronouncing it LibreOffice? Is that right? L-I-B Office, all one word. That was another one I was told. But work together, network with each other too. Okay. Thanks, Caleb. All right, listen carefully. Let's jump into this because it's going to take a while. Lab 2 is one of 2 labs that in person, it's absolute torture for students and me and my lab tech, Joel. Okay. So, but now the torture is more on your shoulders. Sorry, everybody, but listen carefully. I want to stress this. There are several video links I've attached to your document in Word and PDF both, okay, that you need to watch as you go through the lab. Some of these links are the same link posted more than once. Watch it more than once, okay? Including in the lab procedures. The professor who created, one of our professors for ACC created the links, okay, videos, and takes you through what you would have done in person. And they're even kind enough, cards to show you, hey, this is how you apply this formula. This is how you solve this formula. So you know how to make this solution. But it's up to you to watch those videos. Okay? This is not a second lab. I will grade your labs. Okay, and Blackboard lets you know if they're late or not, okay? All right. This lab is a making solutions and dilutions lab. Listen carefully. It's very important to understand what a solution is because after the first exam, we're going to go into this quite a bit. Okay, we're going to talk about solutions inside of your cells and outside of your cells. And I'm going to expect you to understand what a solution is. So simply, I want you to know a solution going into the first exam and after. A solution is made up of a solute dissolved by a solvent. In this class, everybody, what is a solvent always going to be? Not a rhetorical question. Right, water. Okay? So in today's lab, what is a solvent always going to be? Water. Your solute is going to be a chemical called potassium permanganate, KMnO4, and I'll show it to you. Okay? And when you make these solutions, you're going to make these solutions at different concentrations. And remember, I like to use brackets for concentration because it's what they use in chemistry courses. Chemistry, organic chem, biochem, they use brackets, okay, except they should be the same length. All right. Simplest way I can what we mean by the concentration of a solution, well, look what a solution is and then carry it down here. It's the amount of solute you have in a certain volume of solution. Okay? Listen carefully. When I'm trying to explain what we mean by the concentration of a solution, I'm not talking about total volume. I could have two solutions that I compare, okay, and their total volumes are not the same, and yet I tell you they're at the same concentration. And I'm right. And we'll go over this more in chapter 7 in lecture, okay, after the first exam. So hopefully all this will come together. So in your future classes, you have a good grasp. Okay. So after completing this exercise, you should be able to prepare a solution of specified volume and concentration from separate solutes and solvents. That'll be on the video for you. You have a video showing you for the lab procedures. So listen, even though your lab report doesn't require you turning anything in from your lab procedures, the data you need for your post lab is in the lab procedures, and the videos showing you what you've done in person are in the lab procedures. You need to watch those. Okay, I had a student ask me what was the table we were supposed to use for the post lab for lab 1. And well, I said that it was in the recording, and I even pulled up the lab protocol and pointed it out, the table. Okay. Now I appreciate the student asked me the question because they asked it very early on, so that told me they were getting started with their report. But it's in the recording. Okay. Second, prepare a dilution series using both the parallel and serial dilution methods, and be able to determine which method is most appropriate for preparing a given dilution series. So we're going to talk about two ways of creating what I call dilution samples, consisting of a solute at a certain concentration and where the solvent is water. And we're going to talk about how there are formulas that are used for these two types of dilutions to create dilution samples, okay? But the two types of dilutions we're going to talk about, as it says up here, are known as parallel dilutions and serial dilutions. There are formulas that come with each one. And it'll take you through the pre-lab. In the pre-lab, it'll take you through and it'll ask you some questions that don't have anything with calculations. There'll be one section in the pre-lab which will simply ask you, which dilution method should you use, parallel or serial, to set up these samples based on the conditions we've given you. So you have to read the background info to answer those questions, okay? But remember, the lab is constantly feeding into itself. So again, you'll come across pre-lab questions that are prepared that feed into the procedures, and it'll even tell you, rewrite this information in the lab procedures. Even though you're not turning in that section, you still want to do it. And then towards the end of the pre-lab, it'll give you a scenario that's a practice run for what you're going to do again in the post-lab. All right. So now explain what a spectrophotometer is and be able to use one to measure the absorbance of a solution. Explain the differences between dependent and independent variables. That's where the spectrometer, okay, feeds in to this, which is dependent on this. That's given in the background information, and there's some your turn questions to help you practice. Everybody, don't give me haphazard answers. Give me exact, complete answers, okay? In other words, correct answers. Analyze the relationship between two experimental variables using tables, graphs, and linear regression analysis. Whenever you do a linear regression analysis, it'll be with the graph And the information you're going to use to create that graph is always going to be given to you in tables in both the pre-lab. For the post-lab, the tables will be in the lab procedures. Okay, I'll point that out. And then when you do linear regression analysis, that's when you're going to end up plotting what we call a trend line, and then you're going to get an equation. Okay, that equation is simply, in my day, we refer to the equation as a slope equation, representing the slope. Except, as I'll, when you create a graph, you have to know a graph consists of two axes. Sorry. What are those two axes called when you're creating a graph, everybody, before I go any further? What do we call those axes when you create a graph? X and Y axes. Okay. So you're going to use that graph to help you with this. Is that clear to everybody? So I'll tell you, when you create this graphs, the x-axis should represent concentration of your dilution sample. I will tell you the y-axis should represent your absorbance readings from the spectrophotometer. Okay, so I'll talk about that again as we go through it. So a solution is a mixture, usually a mixture, a substance called a solute dissolved, substance called a dissolved, and a liquid called a solvent, in this case water. Any solution where water is a solvent, we call it an aqueous solution. You've heard me use that a lot. Now I'm explaining to you. Anytime I say aqueous solution, it's what I'm talking about. It's a solution created using the solvent water, and that's going to be constant. The solvent is always water. The concentration of a solution describes the amount of solute per volume of solution, as I said before. Concentration can be expressed as a ratio, a percentage, or molarity, but listen carefully. And I'm going to show you this on the next page too, so I'm going to do it twice. Molarity is a unit of measurement representing concentration of a solution. It often goes by the symbol M, or more specifically, when you apply it to a calculation you have to perform, you can rewrite that uppercase M as moles per liter. They're one and the same. They both mean molarity. Now I'm going to give you a formula. Okay, two formulas, and one of those, or three actually total, three formulas, but one of those formulas, the formula, and I'll show it to you again, so just listen. One of those formulas is written down as C1V1 equals C2V2. My students that have had chemistry like to rewrite it as M1V1 equals M2V2 because the M represents molarity. I tell the students the reason why I use C, okay, is because the units for concentration could be molarity, or it could be, this is supposed to be micro, could be micrograms per microliter. Sorry, that should have been the UL. Micrograms per microliter, or, sorry, it's so bad, it's getting worse. Or it could be milligrams per deciliter in this class, unlike chemistry. So just in case, because I usually have at least one student that knows this information pretty good, and I have them sometimes ask me, like this summer, sir, can I use M1V1 instead of M2V2? Okay, but molarity. So this up here, right here. Now, so if you look here, in this class, we use different units of measurement. Okay, so Gomez, stick with what I give you here, okay? We'll save the other stuff for Kim. Remember, we're doing chemistry for biologists, and this is going to be complicated enough. So they talk to you about molar mass and formula weight and yada, yada, yada. What I want to go over is this formula weight thing, because this is what's pertinent. The last line on that previous slide is important for you, but it's what I discussed earlier already. But if you look here, formula weight, what happens is when my lab tech Joel orders chemicals such as sodium chloride or potassium permanganate, on the side of the container, it gives him something like this. It'll say FW, okay? The FW stands for formula weight, okay? This is the formula weight of sodium chloride when he orders it from a company. This is the formula weight of potassium permanganate when he orders it from a company. The importance of knowing the formula weight is that you need it to make a solution from scratch. In other words, I'm taking a certain volume of water and I'm taking the solute, but that's in powder form. So the solute's in powder form, and I'm going to dissolve it in the water. So whenever you're going to make a solution from scratch, you're starting with a solute that's in powder form, and you need to know its formula weight. This is just showing you how they calculate the formula weight. They go to the periodic table, okay? And what they do on the periodic table is they focus on this, the atomic weight. Sodium chloride over here, roughly 23 for sodium. Okay, for chlorine over here, I could barely read it, roughly 35, 36. I'm leaving out some numbers, but round it up. If you add these together, it comes out to 58.5. I know it doesn't come out to 58.5 here, but I left off some numbers. So this is how they calculated the formula weight, by using the atomic masses, okay, or the atomic weights. Potassium permanganate, this is the one you're going to need for your calculations, and they're going to give it to you. They're going to tell you this is a formula weight of potassium permanganate. All I'm telling you is how they calculated it. They go look at the atomic weight for potassium and manganese, and then they look at the atomic weight, okay, for oxygen. You look here at the bottom, atomic weight, you find oxygen roughly 16, except potassium permanganate, okay, what contributes to the formation of this molecule is not one atom of oxygen, but 4. So what they say is take the atomic weight, multiply it by 4. So this is just to let you know where they're coming up with these formula weights. I have some students that basically move on and they work in a lab and they have to make solutions for lab experiments. Okay, and when you get hired, if you haven't already done this, once in a while I have a student who has done it, but if you haven't already done this, they'll tell you, hey, make this solution at a certain molarity, and here you go. And it's up to you to understand, where do I get the information to help me make this solution at the appropriate concentration and total volume? And the formula weight is the key, and it's usually provided on the side of the packaging. In this lab, it's given to you. This is a formula weight of potassium permanganate. These are the units, grams per mole. Why is that important? So that you can make a solution, okay, a stock solution from scratch, starting with the dry solute. Okay, you have to calculate how much dry you will need to weigh out, okay, and dissolve in a certain volume of solute in order to make your solution at the desired concentration. So down here, listen carefully, I believe it's on page 2 of your lab protocol. You're going to need this, and it's provided in your lab. This is the formula for calculating the amount of dry solute that you would need to weigh out. Now in the lab procedure, our professor will demonstrate it for you, what you would have done, because you just don't simply weigh it out. There's more to it, because you want to make sure the amount of dry solute you weigh out, you want to make sure all of that, okay, is transferred into your water. And when you're making a stock solution, the amount of water you use to dissolve that solute, you don't want to use, okay, the total volume. You want to always take whatever volume you want to make that stock solution at. Say you want to make it at 500 mils, final volume or total volume. You want to dissolve the solute in 250 mils of water. Once it's dissolved, then you'll add more water to bring it up to that final volume or total volume that you want, 500 mils. So make sure you pay attention to that also, because in the post lab, it's going to ask you to describe how you prepare a solution from scratch, as if you were teaching it to somebody else, and it's in the video too. So the way we calculate the amount of dry solute that we need, or the grams of solute that we need, because the scale weighs in grams that we use, the electronic balances, is you need the formula weight for potassium permanganate. I already gave you that, 158.04 grams per mole, times the molarity, or moles per liter, of the solution. It'll give it to you on the second page of your lab protocol. It's going to give you a few practice runs, and it's going to give you varying concentrations in molarity. Okay, so you're going to have to take that formula weight that they gave you for potassium permanganate. You're going to have to take the concentration of that solution in molarity, but change it from m to moles per liter so that you can cancel out the units. And then it's going to tell you, make this stock solution at this concentration and this total volume. Now they say total volume, but sometimes some people will say final volume, but it'll say total volume. They may give you the total volume in liters, but if they don't give it to you in liters and they give it to you in milliliters, you need to convert it to liters so that you can cancel out these units. Because remember, in the end, you're weighing out dry solute. So what's going to be the units of measurement when you get your answer? Grams. Okay, that'll be on page 2 with the pre-lab. It takes you through some practice runs, and then it even tells you for one of the 3 or 4 scenarios they give you, take the answer and transfer it to your lab procedure. And even though you're not turning in any lab procedure information, you still need to have it in there to help you with your post-lab. Okay. Then what it's going to do is it's going to say, hey, look, it's going to give you some questions in real about do's and don'ts. You know, hey, I weighed out such and such solute, dry solute, and I realized I weighed out too much. So should I dump it back in the original container, or should I dispose of it in a waste container? And you got to answer. They'll tell you what the person does, and it's up to you to say whether they're right or wrong. And if they're wrong, explain why they were wrong. Don't tell me they're wrong, okay? Then what it's going to do is it's going to transition. It's going to transition into making dilutions, okay, using a pre-existing stock solution. All right, so basically the way it works in person, you would make a stock solution from scratch, starting with dry solute. And then you would take that stock solution at a high concentration and use it to make a second stock solution at a lower concentration. But you don't need to weigh out any more dry solute. Now what you're doing is taking the original stock solution at a high concentration. You're going to take some of that and add it to more water to dilute it down to the desired concentration for the second stock solution you're going to make, which will be at a lower concentration. And it'll tell you in the pre-lab, it'll take you through a practice run. It'll say, sometimes when you want to make a second stock solution at a lower concentration than a stock solution you made from scratch with dry solute, okay, what you can do is instead of making the second stock solution from scratch again, just take the original stock solution at a high concentration, take a small volume of it, but you got to figure out how much do I need to make a second stock solution. And they'll tell you the way you can do that, taking a stock solution at a high concentration and using it to make a second stock solution at a lower concentration. The way you can do that is through parallel dilution. And this is the formula. Again, what does C represent, everybody? What does C represent? Right, concentration. What does V represent? I actually have a lot of problems with V when it comes to units. Volume. Metric system. What is a basic unit of measurement that means volume, everybody? I need to ask this. Right. Okay. But notice the lab. Okay. Yeah, meter cube, but we're going to use liter. Okay. So off the metric system, the basic unit of measurement that directly means volume in its simplest form is just liter. So keep your life simple. That is right, but keep your life simple. Okay. Now, when you go through these pre-labs, it's going to actually talk to you about volumes mostly in milliliters or maybe even microliters. Is that clear to everybody? Okay. But it's here, the V stands for volume. It's also going to talk about to make dilution, make a stock solution, okay, at a very, very low concentration is through a different type of dilution method called serial. Okay. This is the formula for serial, but I'm sorry, everybody. It's not this way on the PowerPoint, but this right here should be shifted over here. This right here, and it's right in your notes. So look in your lab protocol. This right here should be on top. This right here should be on the bottom. I hate this. It happens when I upload it to Collaborate for some reason, but I believe the PowerPoint is right. So in your notes, though, in your lab protocol, though, it's correct. Okay. The formula for serial dilution is dilution factor, or DF, equals 1 plus V2 on top divided by V1. There's no concentration mentioned here. The reason for this is serial dilutions, okay, are formed to simplify the actual preparing of samples. That's one reason when you have multiple dilution samples to create. The second reason for serial dilution is when I want you to prepare a solution, but the concentration and volumes are so low, we don't have the equipment to do it. Okay, so I'm going to go over this. In parallel dilution, it's not just to make, say, another stock solution. You can use it for other reasons, and this is what we're going to go into. But listen carefully. I'm going to say this here. No matter which dilution method you're using, and no matter which dilution formula you're using, we are always solving for V1 in this class. Okay? Always solving for V1 in this class. And again, what is the unit of measurement going to be? Liters, milliliters, or microliters. So parallel dilutions. How can I prepare solutions with specific solute concentrations using parallel dilution? When preparing solutions, it's often easier to dilute an existing stock, notice it says solution, than to weigh out more dry solute. This is especially true if you want to prepare several solutions that each have a different concentration, but the concentrations have to be lower than a stock solution that you have, okay? Now, down here it tells you, C1 represents the concentration of the what? Stock solution. V1 represents the volume of the stock solution that you will transfer, such here, to a new container, okay, to make the dilution sample. Notice it says here, here's my stock. If I transfer 1 ml, it's telling you I'm diluting it, and it's telling you what the factor dilution is. But notice it says dilution. What does C2 and V2 represent? C2 and V2 have to do with the sample you want to make, the dilution sample. Okay, so notice C1 is the concentration of dilution sample in brackets, and V1 is the volume that you want to make this sample at. What's the final volume or total volume you want? I say down here, you're always solving for V1. In this lab, V1 would be milliliters. Now listen carefully. If it seems like I'm skirting through some of these or going through some of these really fast, there are video links that I've attached in the pre-lab directly to parallel and serial and even dry solute. And some of these videos repeat again because they apply to different sections. Keep watching them, everybody. Okay. Professor Ana Maria did a great job. I think she did a great job. She even has note cards. I mean, she's literally trying to hold you by the hand through the video screen, reaching out and hold you by the hand. Okay. But you have to go over this first and then watch her videos and then watch them again. So one way to dilute an existing stock solution is by using parallel dilution. It's showing you the formula here. So listen carefully. In the end, parallel dilutions, we perform this technique when we just need to make one solution and we have the equipment we need to make it using an existing stock solution. Or if we're told, like you're going to be told, to make a series of parallel dilution samples. I believe it's 7. And when they give you the concentrations of each of the 7 dilution samples, they're all going to have the same total volume, but the concentrations are going to be different. And when you take these various parallel dilution samples and you place them in the order of highest to lowest concentration, when you go from the highest concentration parallel dilution sample to the second highest, there might be a 4-time difference in the concentration. But then when you go from the second highest concentration sample they want you set up to the third concentration, there's only a 2-fold difference, okay, in the difference in concentration. In other words, the second highest concentration is only twice as concentrated as the third. So it kind of jumps up and down when you compare one sample to the next, going from the highest concentration in the direction of the lowest. When that happens, you have to perform parallel dilution. So this is just saying more of the same, a different way, so you can look at it here. Come on. And then this is an example, okay? It says, if you want to make 50 mils of a 0.15 molar calcium chloride solution, okay, from a stock, 2.0 mils. So notice they're showing you here. This 2.0 molar is your stock solution. Well, I said C1 and V1 represent your what? Stock solution. But notice over here, 50 mils at 0.15 molar. Well, they're talking about your dilution samples. Okay, so for both parallel and serial, okay, if you see that as a subscript next to C2 or V2, we're talking about the dilution sample. All right, except for serial, we have to skirt the issue with C2, but this is what it means. Then what we do is once you figure out, okay, what concentration or volume, okay, what the specific volume and concentration is for C1, C2, and V2. Okay, then what you do is you next plug them into the formula. So if you've never seen this equation, haven't done it before, what I'm showing here is how to walk it. Okay? We're always solving for V1. So what you need to figure out is what are the concentrations for C1 and C2? What is the volume for V2? C2 and V2, you have to know they have to do with your dilution sample. C1, V1, you have to know they have to do with their stock. Okay? So there should be at least 3 values you have to plug into the parallel dilution formula. C1, V1 equals C2, V2. Okay? So if you look down here, they plug in 0.0 molar times V1 equals 0.15 molar times 50 mils. Now what I tell students to do, okay, is don't multiply this like this. First, what you want to do is take this and actually bring it over. So I need to erase this. They actually multiplied it first, but what I tell students is first bring this here to where it'll read 0.15, if I can do this, molar, 50 mils. And then all that is going to be over 2.0 molar. You can do it either way. I mean, you can do it the way it's shown here. I got this from another professor, but I tell students instead do this, okay, to where V1 is over here. Sorry about that. It should say 1. Now, when you look at this here, you can cancel out the units. Molar divided by molarity divided by molarity. So you can do it this way, or you can first get all the values on one side so you can cancel out the units. And notice all that's left is mils, which are your units for V1. Now, once you've calculated the volume of stock you need, once you've calculated V1, okay, you then need to take into consideration, yeah, but I want the dilution sample, V2, to have a total volume of 50 mils. So now what you need to do is figure out how much water is needed. And it'll ask you this in the pre-lab. So once you solve for V1, what you next want to do is take V1, I'm sorry, let me put it over here. So next you want to do is take V2 minus V1. So now you know how much water to add or how much water is needed. Okay, that's how you'll figure it out. Again, the pre-lab will take you through this, through a practice run here. And so will the professor who gave you the video. There's a video demonstrating this, okay? So I'm going to let you look at that, and I'm going to jump ahead here. Serial dilutions. When do I use serial dilutions? You use serial dilutions for one of two reasons, and it's going to ask you this in the pre-lab, okay? It's going to tell you which dilution method would you use based on the information given to you, parallel or serial. You got to read carefully, okay? But serial dilutions, there's a couple of reasons why you would perform a serial dilution. First of all, you would use a serial dilution when you need to make several dilution samples, okay? And you notice there's a dilution factor, such as, okay, it says here, suppose you have a 2 molar stock solution, okay, and you want to make 15 mils, all right, of 1, 2, 3, 4 dilution samples at concentrations. They're all going to be 15 mils, but they're going to be at these different concentrations. So what they tell you to do, okay, is to take these and arrange them in the order, okay, of highest to lowest. Take the highest concentration, which is actually the stock, and divide it by the next highest concentration, which is here. So you actually have to factor in the stock. You're starting with the stock, okay, and then the next one. But technically speaking, you can get around that, okay. Then what you want to do is take that dilution sample that's at the highest concentration and divide by the next highest concentration. They're telling you, then you would take this concentration and divide by the next highest concentration, get 10 again. You take this third concentration and divide it by the lowest concentration, and you should get 10 again. Okay, so in other words, this is 10 times greater in concentration than this. This is 10 times as concentrated as this, and this is 10 times as concentrated as this. In other words, we have what we call a dilution factor. And whenever you have a dilution factor, you want to perform serial dilutions because it's quicker to set up the samples, but on paper, it's a lot more confusing. So you definitely want to watch the video on this. However, there's a second reason. Second reason you may want to make a dilution sample if I can get it, and I think I have on here. Second reason is what I said, okay? In the sample, the dilution sample they want you to create, okay, requires volumes that are too low that we don't have the equipment to use. Then you want to use a serial dilution, such as the smallest volume that can be transferred in a lab is what's in it better. There are micropipettors that can transfer 2 microliters. Technically speaking, I worked with micropipettors that could transfer anything from a half microliter to 2, but I'm going to tell you right now, there was no way we were being accurate at a half because it's not even a drop of water. Okay, so if the concentration is really low for the sample you're going to set up, you would want to use serial. Now, as I said before, there's a formula for it, and it came out right here. If you're going to set up samples using serial dilution, and it's going to tell you in the lab protocol, it wants you to perform create dilution samples using serial dilution. Whenever you're going to set up dilution samples, create them using this method of dilution, serial dilution. Okay, I want you to notice something. All right, this is the formula you're going to use. Of all, listen carefully. Unlike parallel dilution, I forgot to stress, whenever you're setting up multiple dilution samples using parallel dilution, you have to use that formula C1V1 equals C2V2. You have to perform the calculation for each individual sample. So in the lab, they're going to say set up 7 parallel dilution samples. You have to apply that formula 7 times. With serial dilution, it's not the same. This is the formula, and you only apply it 1 time. Okay, now you're solving for V1. Notice what's missing from here? Concentration. There's no C1 or C2. We don't need it. Okay, what we need is dilution factor. Okay, and I showed you how that can be calculated. And what we need is to know what V2 is. All right, once we have that, then we want to solve for V1. A lot of my students don't understand how to get V1 out from down here. All right, that's why I provided the tutoring link, everybody. It's up to you to know how to do this. Jumped ahead of myself. So what'll happen is it'll give you a stock solution at a concentration, and it'll say, I want 15 mils of each dilution. You'll determine the best dilution method, and if it's serial, you're going to use this, but you're only going to use this formula 1 time, and you're going to solve for V1. Okay, V1 equals. Now, using the previous figure, using here, it's telling you 15 mils. Okay, so that's V2. I'm telling you the dilution factor. So here's 15 mils. 2. Okay, here's our dilution factor, or DF. In the lab, in the pre-lab, what it does is it asks you, is there a dilution factor? And there should be one because it tells you you're going to perform serial dilution. Then it'll ask you, what is V2? Then it will ask you to apply this equation so that you can tell it what is V1. Okay? Well, if you look at that previous page, V2 was 15 mils, and even on this slide. So you replace V2 with 15 mils, and instead of putting in DF, for this example, you would replace dilution factor or DF with 10. So you would rewrite this equation, 10 equals V1 plus 15 mils over V1, and you would solve for V1, and the units, again, should be in. Now, I'm going to let you watch the video in terms of how you set it up, okay? How you set it up. One of the guys you're going to get for serial dilution, the last one is it's going to be confusing, okay? It is possible for V1 and V2 to come out to the same answer, though. But you got to watch the video produced by our professor to understand why it's possible on paper, okay, versus when you actually do the experiment. Now, what happens If you recall in lab 1, we talked about accuracy and precision. Okay. Well, you can determine how accurate and precise you were using statistical formulas, or you can use an instrument called a spectrophotometer. Okay. When you set up your 7 parallel dilution samples, and when you create your 7 serial dilution samples in person, what we would have the students do next, okay, is take each sample, put them in an instrument called a spectrophotometer. That spectrophotometer, okay, has a light that passes, that it'll emit, and we can choose even what wavelength of light. Okay, white light, as they're showing you here, is made up of light particles moving at, simply put, not accurate, but moving at different speeds, resulting in us recognizing it as different colors. But when all of them are together, all we see is white. The spectrophotometer allows you to choose what wavelength you use. The wavelength that we would use in person is a wavelength of light that potassium permanganate absorbs the best. That potassium permanganate reacts with the best. And what will happen is, as this instrument, the spectrophotometer, emits that wavelength of light through your potassium permanganate sample, the lower your concentration of potassium permanganate, the more light that's able to pass through it. Therefore, the instrument will give you what we call an absorbance reading, something like 0.105. Okay, so on the instrument, it'll give you basically, it'll say 0 point something, okay, to 3 decimal places. If you have a high concentration of potassium permanganate, as that wavelength of light enters the sample, more white light or more light is absorbed at that wavelength, less passes through, and therefore your absorbance reading is higher. Okay. Now the way it works for the spectrophotometer, we use it to help you determine how accurate and precise you are. Because if you set up a sample at 1 molar solution, you set up another sample at 0.2 molar solution, and they're at the same total volume, your absorbance reading for the 0.2 molar should not be higher than your 1 molar, first of all, okay? You know you screwed up. So this is what we would do in person. It's harder to do it, you know, this way online. But again, our professor videotaped how you would go through the process, and she'll explain it more, okay? So as I said, now when we take absorbance readings in person, the values we're given will fall anywhere from 0.000 if it's water to 0.200. Usually the readings fall in between. So in the lab procedures, for both the 7 parallel dilutions and for both the 7 serial dilutions, I've given you absorbance readings as if you had done them. And notice the values that I've given you, okay? I've given them. Also notice that the absorbance values I give you, compare them to the concentration of your samples. And see if you notice that the higher the concentration of the sample you created, the higher the absorbance. The lower the concentration of your potassium permanganate in the sample, the lower your absorbance readings. Notice I try to give you something that correlated. It normally doesn't work that way in person, okay? The wavelength you would have used, you would have set the spectrophotometer at, would have been 540, because that's the wavelength of light that potassium permanganate absorbs the best, okay? And here it says, basically, the greater the potassium permanganate concentration of a solution, the higher the absorbance reading. But when you create your graph, okay, when you create your graph, you're going to be using this data. The title you're going to give to the axis that's going to represent these absorbance readings, it's just absorbance 540, but we say it's just absorbance 540, but we say A540. Listen carefully. These absorbance readings are going to represent your y-axis, everybody. Okay? So remember that. Your absorbance readings are going to represent your y-axis. I'll say it now so I don't forget. The concentrations of your solutions, of your dilution samples, when you create your graph, they're going to represent the x-axis. So when you create your graphs, you're going to have, I believe it's 4 graphs total, 1 in pre-lab, 3 in post-lab. When you create these graphs, values always go on the y-axis. Concentrations always go on the x-axis. And when you create your graph, don't just tell me concentration, tell me the units. Okay? So when you label your axes, the y-axis will simply be labeled A540, meaning absorbance at a wavelength of 540. When you label your x-axis creating your graph, you're going to give me brackets, okay, and the concentration. Is it molarity? Is it micrograms per microliters? I'm sorry, micrograms per microliter, milligrams per deciliters, but you're going to give that on the axis. But you'll put Km and O4 on the x-axis and then give me the units of concentration on that axis. So I'll let you read this. It talks about independent variables versus dependent variables. Independent variables are the ones you choose. Well, we're giving you what concentrations of potassium permanganate dilution samples you're going to set up. Those are your independent variables. So your x-axis, which is going to represent the concentration of your potassium permanganate samples, that's your independent variables. The other variables are your absorbance values, A540. The reason why they call these dependent variables is because the absorbance is dependent on the concentration that you set up for your potassium permanganate. Okay, and that is plotted on the y-axis. So notice what I'm telling you here, everybody. Everything I just said before is right here. Okay. Graphing data. Graphs are visual representations of data that often bring out relationships. They can have many possible formats. In this course, you will be graphing your data as scatter plots. So in the pre-lab, it's not going to say create a graph. It's going to say create a scatter plot. Sometimes it'll say graph, but if it says scatter plot, okay, remind yourself that means it's a graph, everybody, that you're creating. Okay? You do not connect the dots. When you create a scatter plot, it's going to show both your A540 and concentration. Where they're related, it's going to come out as individual point circles. You do not connect the dots. It's a scatter plot. You don't connect them. I shouldn't see a wavy line connecting your data points. All right? But what you will do is you'll do a linear regression analysis. I got to remember to post some tutorials that may or may not help. I don't know if they may be outdated. Then once you create that scatter plot, okay, if you're using something like familiarize yourself, that's on your shoulders, but I'll put up those docs after class, see if they help still. Or you can go in for tutoring and they'll show you. And it'll ask you to perform a linear regression analysis in both pre-lab question and in the post-lab questions. Okay, this linear regression analysis will show up on your scatter plot graph. It'll be that best fit line running between your little data points or circles. Then you will ask the program, show me the linear regression equation, and it's going to show you an equation. But listen, it's going to give you a numerical value for the slope, and it's going to give you a numerical value for the y-intercept. Now listen, in the lab procedures, look at the table I gave you that contains the absorbance values for the serial dilutions. And I'll show you this again on the doc. The very last row, it says unknown, and I gave you an absorbance. You do not graph that unknown. That unknown you're going to use for the last question in post-lab. Okay? For that unknown, what I'm giving you is the concentration of the unknown. Okay. I'm sorry, I'm giving you the absorbance value of the unknown. I'm giving you the absorbance value of the unknown. So when you take that value I gave you for the unknown and you apply it to this equation, which it'll ask you to do in question 6, you have to remember absorbance values are represented on the y-axis. So the y and the x here are associated with those. And it'll say in the last post-lab question, calculate the concentration of the unknown using the information given. You will be given the absorbance. What you will solve for is x, and the units of measurement, the concentration units of measurement, look at your graph. That's how you know what the units of measurement is going to be. I talked with our chem professor, Professor Allison, and students have to create a lot of graphs, and he tells me students don't know how to read graphs, and it's not about knowing it, it's about learning how to do it. And he says, I can't even go through it baby steps, and it's a struggle. But listen, when you create these graphs are going to be used to answer other questions, especially in the post-lab, or to solve for your unknown concentration. So listen, pre-lab, your turn. Questions and answers. Show all your working conversions. Listen carefully. You don't have to show all your work. Just show me how you set it up. and I'll take that as enough. Is that clear to everybody? That's fine. Conversions, that's up to you. So make sure you listen to this again when you're doing it. You don't have to show me all your work since this is virtual or online class. Just show me how you set up the problem. Give me the formula and then plug in the values with their units of measurement, and then just give me what V1 equals, say. Or give me how many grams of solid. That'll be good enough, okay? So you'll answer all the pre-lab questions like before. Same way that you did lab 1 is how you're going to handle it here, okay? Except you have a lot more calculations, I believe. Post-lab, there are 6 questions. You're going to answer all 6. Please listen carefully. Question 3, the department designed it to where they wanted you to take the results for the parallel dilutions, create a scatter plot, take the results for the serial dilution, and create a scatter plot overlapping the parallel dilution sample. I don't care for you to do that. I found it works a lot better for students because, again, as I said before, I've talked to our chem professor about being able to read graphs, and I don't recall if I edited it, but just create 2 separate graphs. Okay, one graph for parallel dilution data and a second graph for serial dilution. Okay, and when you do this, I want you to do a practice run. Ask it to do a linear regression analysis and give equation, and then give it the R value. Listen carefully. You're going to need the R value for both graphs, okay, for question number 4. Make sure when you read the PowerPoint, sorry, it should be a Q. Make sure when you read the protocol, and I have it back in the PowerPoint, it basically is telling you the R value gives you an idea of which set of data, the set of data used to create your parallel dilution graph or the set of data used to create your dilution graph. Which set of data do you feel demonstrates which of the dilution techniques is more, more dependable? Are parallel dilutions more dependable as far as accuracy and precision, or are serial dilutions better to use because they're more accurate and precise when you want to set up samples? You'll use this off these 2 graphs. Then what you're going to do next is you're going to get to question 5. Question 5 here, it's going to tell you combine your data. So this is going to be tedious. What you're going to do is go to the 2 tables that I've given you in the lab procedures. You're going to take your parallel dilution concentrations and absorbances. You're going to take your serial dilutions, concentration and absorbances, and you are manually going to have to rearrange them in the order of highest to lowest concentration, okay? I'm sorry. That's why I put this here. You're going to rearrange them from lowest to highest concentration, okay? So you're going to have 7 for parallel, 7 for serial. You're going to rearrange them from lowest to highest concentration. Now, this is going to be perfect, but it's good enough. Then what you're going to do is you're going to create a scatter plot, ask it to do a linear regression analysis, and ask it to give you the linear equation. And then you're going to take this right here, this equation, and you're going to use it for question 6. Question 6 is going to tell you, calculate the concentration of your unknown sample. The absorbance for the unknown sample, again, it is recorded on the bottom row where I gave you the concentrations and absorbances for the serial dilution samples under lab procedure, and I'm going to show you this, okay? All right, so let me go over to the, welcome to 1406, everybody. Oh, come on. I'm sorry, I'm going to start cursing here. Try not to use bad words, but I get frustrated when it's not working here. Okay, let's see if it'll work with me here. Okay, so this is page 1. Make sure you read everything here as you go on into the second page, if I can get it to go to the second page. Okay, this is where you'll get your first set of questions that have to do with calculating the amount of dry solid you use. There's a video link right here. Make sure you watch. You'll see that video link multiple times. Here's the formula that you'll need that I gave you on the PowerPoint. It's a little more simplified here, but you got to compare the 2 because a lot of students don't pay attention to what they read and they don't realize moles per liter is molarity, okay? It'll give you some practice runs, and then on one of them, this one here, it'll tell you to transfer that data to the lab procedures. Even though you're not turning in the lab procedures, transfer the data because you still need to go through the lab procedures to answer the post-lab questions. Then what you'll do is you'll move on to the next page, okay? And on this page is where it'll give you scenarios about do's and don'ts. What did the person do wrong? And why did they do it wrong. So you got to give a little extra description. Okay, and then it'll get into parallel dilutions. Watch the video link. Our professor goes to help you out. This is where it gives you the formula, and then it starts talking about what each of these variables C1, C2, V1, V2 represent. But watch that video as you go through this, please. Okay. Then as you move on, you're going to get to your next set of pre-lab questions in your term box. Okay. I put the video link again. That's reminding you, hey, you're probably going to need to watch it more than once. All right. Don't send me an email saying, oh, I don't understand this, and then you never looked at the video. You got to watch it multiple times. Then after that, you're going to use that formula to answer the question here. Make annotations. When you're reading the question, write down what unit of measurement, okay, or what volume represents V1 or V2, which concentration represents C1 or C2. Make those notes up here to help you out, okay? And then it'll tell you transfer that information. But it not only asks you to solve for V1 using the parallel dilution formula, then it talks to you about water needed. V1 minus V2 minus V1 equal the amount of water needed. And it'll tell you to transfer some of that data to lab procedures. Make sure you do that. Okay. Then you'll move on to serial dilutions. Okay. Serial dilutions, there's another video here. Again, watch that video. It's first going to ask you some questions about, based on this scenario, based on the volume and the concentration of the sample we want you, or stock solution we want you to, which is better, serial or parallel? And then it'll say explain why. Okay. Then what it'll do is it'll say, okay, now that you've done that, we're going to move on, and now you're going to use and apply the serial dilution formula. Everybody, I'm circling the serial dilution formula. Sorry about the spacing, but this is all one formula here. Then what it's going to do on the next page, after it goes over the background info on this, because the way I say it may not be good enough, you may need to see it again, gives you a practice run, and then I post the same video again, letting you know you need to watch that video more than once. And then you'll enter some questions, your turn questions in the lab, and it's going to say, what are the dilution factors of the samples we've asked you to set up? But it's telling you you're going to use serial dilution, so you know there has to be a dilution factor. Then it's going to say, what is the volume of V2? Then it's going to say, calculate V1, and then it's going to give you this last question, when I'm going to leave it up to you. It's a thinking question. Okay. Now, it seems simple, but usually that's the one that does not know how to solve for V1. Then it's going to get into the spectrophotometer, read through this, it'll get into the R value, get into linear regression, it tells you everything background that I gave already. Talks about this equation. When you go over this equation, make sure you go over everything with this equation. It's telling you, okay, because I skimmed over everything. Up here, it's giving you scenarios, and you have to identify what is the dependent versus the independent variable. Okay, so remember, if it's a dependent variable and you have to create a graph, it's represented on the x-axis. I'm sorry, if it's an independent variable, it's represented on the x-axis. If it's a dependent variable, it's represented on the y-axis. Then they give you a practice run. It's going to give you data. Listen carefully, everybody, for those of you that are still here, okay? This column here, it's giving you protein concentration that you're going to graph. Which axis would you put this data on? X or Y? Protein concentration. Right here on this figure, where it says protein concentration, okay? Yeah, X. So what you want to write in the box then is x-axis to remind yourself. Over here, it gives you the absorbance reading, and even though it's not the same absorbance reading you use for potassium permanganate, okay, yes, it's the independent variable. Even though this absorbance reading is going to be different than the one for potassium permanganate, it's still going to be plotted on which axis for the absorbance? The Y. So make little notes and annotations for yourself as you do this, okay? Write in x-axis, write in y-axis. This is taking you through a practice run for what you're going to do in the post lab with the data I gave you in the lab procedures. Okay, it's going to ask you a couple of questions, and then it's going to say create a scatter plot. Remember, that's a graph. So you're going to create a graph. You're going to perform a linear regression analysis. It's going to give you the slope-intercept equation. You're going to do the linear regression analysis, and then you're going to ask it to show you the linear equation, the trend line equation, y equals mx plus b. right about here. Then you're going to take that equation and you're going to apply it here to solve for the concentration of the unknown, because they're going to give you the absorbance. Okay? And remember, when you use that regression equation, or trend line equation, y equals mx plus b, y represents the y-axis, which is the absorbance readings. Okay? In that y equals mx plus b, x represents the concentration, the independent variable. In your graph, when you create the scatter plot graph, you not only have to put down this protein concentration, but you have to put down what is that concentration. That also is part of the title of the x-axis. That way, when you solve for the concentration of the unknown down here, if you don't know what the units should be, look at your graph or this table. It tells you what the units should be. Okay? That last pre-lab, that second to last pre-lab, that's actually what you're going to be applying in the post-lab. So it's giving you a practice run. And then it just asks you to rewrite the formulas that you used at the beginning. How do I calculate grams of solutes, parallel, and serial dilution? Now here in the lab procedures, I'm going to jump real quick here. This is where you're going to find your first table, okay? For parallel dilution, this data and this data is what you're going to need to plot. I gave you this. Normally, you would have to perform, okay, or apply that parallel dilution formula 7 times to figure out how much V1 you were going to add, and then figure out how much water you would add. So I gave you that link again to watch. Okay, you're lucky. You don't have to go through all this. Most of my students can't even calculate how much V1 and water can do this, unfortunately, but it's good practice for them. It's okay to make future courses easier if you're going to have any that have to do calculations. But this is your concentrations. These are your absorbance for your parallel dilutions. You'll create one graph in parallel in question 3, and then this is the data you'll combine for question 5 with your serial dilution data. But listen carefully. The serial dilution data is just this. What I'm trying to put a box on, okay? These 7 top concentrations and 7 absorbances are what you're going to use to create your serial dilution graph for question 3. And then these are the 7 pairs of data that you're going to combine with the 7 pairs of data for parallel to create that even larger graph for question number 5. Okay? What you're not going to graph at all, what you're not going to graph, you never graph this, is this. We were just trying to find a spot to place it so students would find it. Okay? This right here is the unknown, and this is the absorbance of the unknown. You are going to use this data here to help you with calculate the concentration of your unknown in question 6 using that graph you created, and that's y equals mx plus b equation that was derived from question 5. Again, there are links here for the spectrophotometer and what have not. Make sure you watch these, everybody. Many of us in the biology department, from what I can tell, I know I'm one of them definitely, really want to get back in person, because as far as I know, ACC is one of the only places where you're going to come across professors, first of all, teaching in the lab. Second of all, I know some like myself, we actually want you to learn something in the lab. A lot of our students appreciate it. So these are your post-lab questions, okay? You have 6 post-lab questions. All right, and here's question 5 and 6. We're at 5 where you're going to combine, okay, all 14 sets of data combined from your parallel and serial to create a graph. Once it does a linear regression analysis, it gives you that trend line equation, y equals x plus b. Then you're going to use that equation plus the data I gave you at the bottom of the serial dilution table for the unknown absorbance, and you're going to use that to calculate the concentration of the unknown. Now, if you don't know what the units are for that concentration, all you'll do is go back to your graph here. What did you put down as the units of concentration for the concentration on the x-axis? That's what lets you know. So the graphs are not busy work, okay? You're creating graphs to help you analyze your data and extract information, okay, from that. such as so that if one day you end up doing work where you have to do that, you understand how to process and analyze your data and extract, okay, conclusions, or data results that will feed into your conclusions. Okay, yes, this is I didn't design this lab. That's my disclaimer, okay? But I do expect y'all to learn how to do it. It will pay off in the long run, okay? All right, any questions? If not, just listen to the recording. Watch the videos. This is going to take time. Do not wait till the last second, everybody. Okay, then I'm through with the session. I'm going to stop the recording. I'm going to get on it, posting those tutorials for Excel. I think